Okay, we're continuing our lectures in chapter 3 by now looking at the class of organic macromolecules, the lipids. Remember that one thing that separates the lipids from the other three groups was that these are not polymers. They're grouped together because they are hydrophobic, and recall that that refers to them having little or no affinity for water. Now, just because they're not polymers doesn't mean that we are not going to see them being synthesized through dehydration synthesis or being cleaved into smaller pieces using hydrolysis. It just means that they're not built by adding monomer to monomer to monomer. Now, there's three classes of biological lipids that we're going to explore: fats, phospholipids, and then steroids. If we look at fats first, fats consist of a glycerol and fatty acids, and these fatty acids are usually long hydrocarbon tails full of carbons and hydrogens that are formed by dehydration synthesis. So we can see here when we add this hydrocarbon tail, this fatty acid, to our glycerol molecule, we move off this molecule of water. Now, one of the main functions of fats is that these are very efficient at storing energy. In fact, per unit weight, they can store about 2 times the number of calories or amount of energy as a small sugar molecule. A secondary function in animal tissue of this is, in addition to storing the energy, the fat itself, because of its hydrophobic nature, tends to form these globules in our cells and makes them sort of blobby or cushiony. And this means that adipose tissue is very, very good at insulating or cushioning things. Now recall that adipose, the word, ends in OSE, but this is not a sugar. It's the exception to the rule there. Now we can break fats down into two categories: saturated fats, in which the fatty acid tail does not have any double bonds in the hydrocarbon chains. Recall that these illustrations of the fatty acid tails denote these carbons and hydrogens here. In a saturated fat, there are no double bonds. Remember that carbon forms 4 covalent bonds, and there's no double bonds joining carbon to carbon atoms. So that makes these hydrocarbon chains, these fatty acid tails, very linear, very straight, and very stable. Their stability makes them more difficult to break down, and so these are things that are solid at room temperatures. So things like most animal fats, like lard and butter. Now, of course, one of the themes this semester is the link to a lot of the topics that we're talking about in cardiovascular disease and heart attacks. So, a diet that is very, very high in these saturated animal fats can tend to increase the risk of cardiovascular disease in this condition called atherosclerosis, which is where some of these these saturated fats can be deposited in the walls of your arteries, causing blockages or occlusions that can lead to heart attacks or stroke. Now, unsaturated fats have the fatty acid tails where we see double covalent bonds in these hydrocarbon chains. And if we look at this illustration, we can see a saturated fatty acid, a saturated fatty acid, and then here is an unsaturated fatty acid tail. Notice that this double covalent bond is denoted by these 2 dashed lines here, gives this molecule a very crooked appearance, and this weakens the molecule's stability somewhat from a breakdown and hydrolysis standpoint. Now, it means that plant oils like olive oil or corn oil are liquid at room temperatures because they're not quite as stable. Another category of our lipids are phospholipids. Now, these are similar to fats in that there's a glycerol, but there's only 2 fatty acid tails. And in addition to the glycerol and the 2 fatty acid tails, there is a phosphate group attached. Now, remember, the phosphate is the functional group with a phosphorus atom and 4 oxygens around it. Now, the fatty acid tails being extremely nonpolar, and this phosphate group with all of these oxygens around it being polar gives these phospholipids a very unique set of characteristics. In fact, they have both hydrophobic and hydrophilic properties. The word that we use to describe a molecule that has both hydrophobic and hydrophilic properties to it is amphipathic. The word amphipathic refers to both, right? So it's both hydrophobic and hydrophilic. Now, these phospholipids are really important because when we place them in an aqueous solution, water, they orient themselves with the hydrophilic heads, these round balls, which contain the phosphate, towards the water, and the hydrophobic tails rotate in towards each other. Remember, the water-hating hydrophobic tails and the water-loving hydrophilic heads. Now, this phenomenon is called the phospholipid bilayer, which is seen when we place these phospholipids in water. And this particular phospholipid bilayer will become very important as we move into our second unit when we start talking about the cell, because the phospholipid bilayer is, of course, the basis for cell membranes. All right, finally, we've got some steroids. Now, steroids are not just things that help athletes to become muscle bound, right? Although they are important hormones in our body that occur naturally, like estrogen and testosterone, cholesterol, which is found in all animal cells, is also a steroid. Now, these steroids have many different properties to them, but they are hydrophobic and do not like water. It's one of the reasons why some of these performance-enhancing drugs, the illegal substances, are often applied as creams because they can be dissolved in these hydrophobic things, and they can actually penetrate through the surface of your skin because of their hydrophobic properties.